اوكي، السلام عليكم. رح نحكي بهاي الليكتشر إن شاء الله عن some definition about biopharmaceutics and pharmacokinetic. ورح تلاقوا إنه إحنا طول الكورس رح نكون نحكي عن ال biopharmaceutics as BP and pharmacokinetic as BK. اوكي، أو زي ما قلنا هذا الكورس هو رح يكون ال 4456 على جهتين، طلاب صيدلة ودكتور صيدلة على خير إن شاء الله. هلأ بالنسبة ل biopharmaceutics it's a part or a science of two kind of things. One that is related to bio and the other one that is related to pharmaceutics. Bio, from the name, it's related to biology or biological environment. Pharmaceutics is the science that you already studied in 1 and 2 and 3, which, if you remember, it's related to the drug itself, related to its properties, dosage form, route of administration, its delivery. So anything that is related to pharmaceutics that you already studied before is involved, but it's not only that, it's the interrelationship or the contact point between the biological system or physiological environment that you have and the drug. So the science that studies this two kind of things, or the relationship between your biological system or physiological system and pharmaceutics, is biopharmaceutics. So anything that is related to both of them and the relationship between both of them will be under this science. As a definition by the American Pharmaceutical Association, is the study of the factors influencing the bioavailability of the drug in man and animals, and the use of this information to optimize pharmacological and therapeutical activity of drug products. Again, we said it's the effect of the drug or the relationship between the drug and everything that is related to it to the physiological system, or whatever the effect of the physiological system to the drug. Okay, so it's any factor that is involved under this umbrella is biopharmaceutics, but usually they consider it more important in the bioavailability of the drug. And again, it's in the physiological system, whether it's in a human or animal, and how we can use this kind of info to optimize the final target that we want from the drug, which is the pharmacological effect of the drug. So again, it's related not only to bioavailability, is usually related, we will talk about this later, to the extent and rate of the drug reaching the systemic circulation. But it's not only related to that, but mainly because it's one of the major things that we want to get the final effect of the drug is the bioavailability of the drug. The factors that could involve, as I mentioned, this is just an example or examples of whatever is involved in this, but it could be anything related to the drug and its relationship to the physiological system. So the structure of the drug, the nature of the drug, excipients that we are using, how we manufacture, physicochemical properties, and many other factors. How we do the biopharmaceutics studies, we have usually, of course, we have different kind of studies or ways to study it, but one of the most common studies is the in vitro studies and in vivo studies. And I'm pretty sure that we know what is in vitro and what is in vivo. When we are talking about in vitro, we are talking about studies that we are not using lab animals or humans. Okay, many students ask me about this before, considering this as without using lab. No, it's without using lab animals or humans. It's just laboratory abbreviation. So anything that we do outside the living bodies or living organisms, yes. So if we are talking about in vivo, we are talking about anything that we do inside the living human beings or animals, whatever the model that we are using. So we could either do the in vitro studies, or in vivo studies, or both of them in collaboration to understand what is happening in the biopharmaceutics of the drug. For pharmacokinetic or PK, we have two kind of things. We have pharmaco and kinetics. Pharmaco, we know it's an abbreviation for pharmacon, as any science that we have in pharmacy. It's pharmacon, which means drug. Kinetics is the movement or motion of the drug. So we are studying anything that is related to the kinetics of the drug in the body. So we are just studying the kinetic of the drug. Okay, and we know, as you already have this in almost every course that you have in pharmacy, the ADME, which is the when they ask you what is pharmacokinetic, you'll answer directly it's ADME, which is absorption, distribution, metabolism, and excretion. Okay, so the science, because it's the main process that is happening of the for the drug in the body. So the main things that we study in pharmacokinetic science is the absorption, is the distribution, metabolism, and excretion. Okay, when we are talking about metabolism and excretion, we are talking about what we call as elimination process. So both of them, the metabolism and excretion, is elimination process. And when we are talking about elimination and distribution, we are talking about what we call it disposition of the drug. So those are like general definitions or general terms that we will frequently use in this course. You already know some of them, and we will again define them in this course because I want you to know what exactly those terms are. Again, it's the science to study absorption, distribution, metabolism, and excretion. We have elimination that includes both metabolism and excretion, and we have distribution and elimination, which we call it drug disposition. When we study kinetic, usually we do the biopharmaceutics part of the study. We understand some of the things that is related to the drug, and we expect what we will have in the body. Then we will do the pharmacokinetic study, which includes two main parts: the experimental part and the theoretical part, or the experimental methods and the theoretical methods. When we are talking about experimental part, we are talking about sampling, whatever the samples that you need to take. We are talking about the techniques to take those samples. We are talking about the analytical methods to analyze those samples for the drug and its metabolite, if you are interested in the metabolite. In addition to the theoretical models or theoretical methods that you are having. After you are taking your samples, after analyzing those samples, you'll have data. So you need to do some kind of modeling to have the models. So we will have kinetic models, and then we will do statistics on them. Let's talk about this a little bit on this graph. We have, again, you'll find so many graphs in kinetic and profiles, but once you understand the model, you will be okay to understand what we are talking about. You will find a lot of concentration versus time profiles. So we have concentration, let's say it's a plasma concentration of the drug versus time. And this is after oral administration of drug X, okay? We find here those dots. Those are usually your experiments. After you do an experiment on animals or human, you will have different samples at different time points. Then again, you will need to understand how you will take those kind of samples, why you want to take those kind of samples, the sampling techniques, the analysis techniques, and then you'll have different data or information of the concentration of the drug versus time. Okay, so this is the experimental part of your drug or your study. Okay, let's change the color and let's talk about this part, which is what the profile that you have. This is what we call it the theoretical part of the the study, or what we call it, the model. Okay, so this is the theoretical or the mathematical part of the model. You are using whatever that you have from the data that you took from your animal or human, and then you'll have some kind of math to model those data to get some kind of relationship between them, and to get your model. And after you have your model, for sure you will have some kind of statistic to compare this profile, let's say, and under condition A, from other profile under condition B, or the drug under, let's say, healthy condition versus the drug, the same drug under disease condition, or the drug, if it's oral, under the drug if it's intramuscular, let's say. So we need to do, as we mentioned here, after the modeling, we need to do some statistic to understand which is better and which is significant different or not. Okay, so this is like the general definition of the pharmacokinetics. Again, it's the science that we study different kinetics or transport methods or transport pathways, but usually we just call it kinetics. It's the scientific term for it. So we study the kinetics of the drug under different processes such as absorption, distribution, metabolism, and excretion. When we are talking about metabolism and excretion, it's elimination. This is important to know. And when we are talking about distribution and elimination, we are talking about disposition of the drug. Okay? The exact definition of each one of those is very important, because usually you consider yourself know what is each one of them, but some kind of tiny details slip from your mind, so we need to redefine them to make sure that you know all of them. When we are talking about absorption, we are talking about the process by which the drug, let's consider this the body of the human, let's say, okay, it's just a sample of your body. If you are taking the drug by oral route of administration, so the drug that you have needs to pass through different kind of membranes, right? So passing through this membrane or different membranes, let's consider those our membranes, whatever the membranes that we are talking about. If we are talking about oral, mainly we are considering the GI, but we may have different other membranes. But it's the process by which the drug proceeds from the sites of administration to the site of measurement. At this point, the site of measurement would be what? Your, let's say, blood or systemic circulation. So the movement of the drug from the site of administration to site of measurement, at which we are taking different samples to do our study. This is what we are calling absorption. Okay. When we are talking about distribution, again, I will consider this two other sites. Okay, if this is the first site that we're talking about, distribution is the reversible, this is very important, reversible transfer of the drug from the from into the site of measurement. So we have other tissues. Let's consider it as other tissues or organs. It could be one, it could be two, it could or could be different organs. So it's the movement from into the site of measurement. Okay, when we are talking about metabolism, then we are talking about the process that is irreversible, changing of the drug, which is the parent compound, completely to a different species. So we are changing, let's say, A irreversibly to another different, completely different entity to B. And this happens by different chemical interactions that we know about metabolism, phase 1 or phase 2. Okay, but you need to know that it's irreversible change of the drug parent drug to other compound. okay, or other different kind of species or entity. When we are talking about excretion, we are talking about the irreversible loss of the drug unchanged from the body or unaltered from the body, which means that I'm losing the drug as it is. This is the excretion of the drug. And again, it's irreversible loss of the drug. The question that we usually ask ourselves, do we have an excretion for this, which is the metabolite? Let's say metabolite, we have one metabolite for the drug. Do we have excretion for it? In other terms, do we have irreversible loss of the metabolite from the body? Yes, we have. And again, this will be the excretion of the drug, and this will be the excretion of the metabolite. Does this mean that this is metabolism? No. Once you are losing the X, whatever it's the drug or its metabolite, unchanged irreversibly, we're talking about excretion. Okay, so the whole process here is in a blue is called excretion. The whole process in here is called metabolism. Okay, so we have excretion for the metabolite, but we have it already metabolized from the parent compound. So it's an excretion of the metabolite. Okay, sometimes you'll have it in your textbook or your reference books as excretion or the excretion of the drug and its metabolite. It's true, it's an excretion of the drug and its metabolite. But again, when we are talking about metabolism, it's irreversible change of the drug to or any entity to different other entity. When we are talking about excretion, it's the irreversible loss of the drug unchanged, okay, out of the body. When we are talking about both of those processes, let's say we are talking about this, which is the metabolism, and we are talking about this, which is the excretion. We call it elimination. So the process of the irreversible loss of the drug or any other chemical compound that we are interested in outside of the body, whatever is unchanged or changed, it's elimination. Okay, so those again, you know most of this, but it's important to just emphasize those terms. I will stop here and we will continue in the next video, inshallah.